CorExplorer finds tumor-associated gene expression factors in a way that is mathematically principled, using interactive visualizations as well as clinical and database information for biological discovery.
CorEx performs information maximization in a way that requires relatively few samples to find clusters in high-dimensional data. The hierarchy of latent factors that it produces facilitates biological understanding. This method has already proven interesting when applied to cancer biology. Its information-theoretic foundations make it applicable to any system with many variables and little prior knowledge about their relationships.
Begin by navigating to the CorExplorer homepage. Under Quick Links, click on the plus expand button to see a summary of the CorEx factor graph that was trained on the cancer data of interest. For example here, the factor graphs for TCGA ovarian cancer data are shown. After inspecting the factor graphs, click Lung TCGA LUAD to access the CorExplorer page for lung cancer RNA sequencing and use the CorExplorer factor graph window to explore the CorEx factor graph for a gene of interest. While moving the mouse cursor over the factor graph display window, zoom into the factor graph using the mouse trackpad to view the details of the graph, such as the most important genes in each factor and the connections between the nodes at different layers. To locate a target gene, click the Gene menu and type the gene name to select it in the drop-down list. Press Return on the keyboard to make the view zoom to the factor with which the gene of interest is most strongly correlated. Reposition the mouse over the graph display and scroll to zoom out to see the level of the node and its associated factors that are neighbors to the most closely associated gene factor.